which are resistant to pyrethroids, the cuticle thickness is extremely high. That is one study I have uploaded yesterday. I have uploaded that paper in that. And another thing what they have observed is, I will take a picture of that. Okay. See, this is the left side one is the susceptible one, and the right side one is the resistant one. See, this is the susceptible one, and this is the resistant one. See, these are the enocytes. Enocytes are the epidermal cells which secretes the epicuticle. See the case. These are the ones. See this reddish colored ones. These are the genes, cytochrome P450 genes responsible for the formation of this phospholipids. And this gene, see in this susceptible one we have only one gene. And see the case over here, we have four genes, four copies of genes. That means the phospholipid composition can be enhanced four times over the susceptible one. Instead of a single gene, this is just a diagrammatic representation. Instead of the presence of a single gene in susceptible individual, in resistant gene, the epidermal cells has more gene copy, which leads to the synthesis of more quantity of the cuticle components, and it will lead to the thickness of the cuticle. See over here. See, this is the susceptible one. This is the resistant one. See the thickness. Resistant ones are having greater thickness of cuticle compared to the susceptible one. So the cuticle thickness increases. There can be an alterations in the gene which will help enable insects to increase the cuticle composition. Okay, are you clear with this increase in the cuticle thickness?
Yes, sir.
Yes. The next mechanism most of you might be very familiar with, that is one which is usually taught to students also. Increased detoxification because of the presence of more detoxification enzymes. This enzymes which detoxify any toxin, any plant. Say, take the case of any insect. You might have seen insects which are feeding on neem, insects which are feeding on tobacco, Spodoptera vetura. We call it as tobacco caterpillar because it has the ability to feed on tobacco. Termite, Heliophiltis, tea mosquito bug, they feed on neem tree. Neem contains acid reactive, but still these insects are able to feed on it because they have the ability to detoxify this uh, allelochemicals that or secondary metabolites present in the plants. Every plant has one or the other secondary metabolite. Take the case of cucurbits, they have cucurbitacin. Only those insects which have the ability to detoxify cucurbitacin can feed cucurbits. So this is the peculiarity of each plant, and the insect has developed ability to detoxify that, and that detoxification is being carried out by enzymes such as cytochrome P450 monooxygenases. It is also called as mixed function oxidases, MFOs. It can be due to esterases or it can be due to glutathione transferases. These three enzymes, glutathione transferases, esterases, and cytochrome P450 monooxygenases. These three enzymes are present in all insects, all organisms. And these enzymes are responsible for catalyzing reactions which will help to detoxify this any xenobiotic, any chemical. So if it is an insect, if it has to consume a plant, it has to detoxify the metabolites, secondary metabolites present in the plant, allelochemicals present in the plant. Then they should possess these enzymes to detoxify these allelochemicals or secondary metabolites present in the plants. So these enzymes are there already present in an insect. Otherwise, insect won't be able to feed any plant which has a diverse array of the secondary metabolites, diverse array of allelochemicals. Insects possess that chemicals which protects them from insects, but insects possess these three enzymes which will enable them to detoxify these allelochemicals. So this is how. Have you heard about insect plant coevolution?
Yes, sir.
Gene
Yes.
to gene,
Huh?
Yes, sir.
gene to gene interaction.
Gene to gene. Oh, okay. Have you heard about any the scientist who coined the term
Plank.
insect plant coevolution? Mail give that paper to you, the classic paper they proposed this. That means this is a constant war going on between insects as well as plants. Once the plants evolved, once the insects evolved, insect tried to feed on the plants, plants tried to resist the insect attack by synthesizing uh, metabolites or compounds which can reduce the insect attack. Insect tried to overcome this by developing enzymes which can detoxify. So this is how this warfare is going on between insect as well as plant. So these enzymes are already present to detoxify a pesticide. That's the only thing they do. So these enzymes are present in insects which will help the insects to detoxify pesticides. And if two pesticides having same metabolic pathway, take the case of pyrethroid. Chemically, all the pyrethroids are esters of acids and alcohol. You might can you name the acids present in a pyrethrin, natural pyrethrin? What are the two acids present in a pyrethrin?
Jasmonic acid,
First,
chrysanthemic acid.
xanthamic acid and
Chrysanthemic acid.
uh, chrysanthemic acid and pyruvic acid. Okay. Then what are the alcohol?
Jasmine.
Cinnarolone, pyrothrolone, and
Oh,
jasmolone.
Jasper.
Okay. These three, these two acids and these three alcohol molecules, they can have six different combinations, and we can have six different kinds of ester bond we have. Acid and alcohol react to form an ester bond. So we can have pyrethrin 1 and 2, cynarin 1 and 2, and jasmolene 1 and 2. Okay. So these are ester bonds. So any uh, enzyme which can detoxify this ester bond can detoxify pyrethroids. And if any other group of insecticide have ester bond in them, this same esterase enzyme can detoxify the OP insecticide also. I will repeat. Esterase is an enzyme which can break ester bond. I told you pyrethroid insecticides are having ester bond. So if one insect has evolved resistance to pyrethroid, it will have high level of esterase enzyme. So this esterase enzyme will break the ester bond of pyrethroid insecticides and insect will evolve resistance. So take the case of OP. OP insecticide, most of the OP insecticides are also esters of phosphoric acid. You might, if you go back to your PG notebook, you can see the OP insecticide in the once they are discussed. First, they will discuss about phosphoric acid. So these are also OP insecticides are also esters. So esterase enzyme, their function is to hydrolyze any compound with an ester bond. They don't differentiate between pyrethroid or OP. Actually, it is a higher level of esterase occurred because of the continuous use of pyrethroid. So the farmer discontinued pyrethroid and used an OP insecticide. Again, that OP insecticide is having an ester bond. Esterase, it doesn't differentiate OP and pyrethroid. Only it looks for is an ester bond. An ester bond is present in an OP insecticide, and it breaks down that ester bond and breaks down that molecule. So what kind of a resistant mechanism is it? Cross resistance or multiple resistance?
Multiple.
Multiple resistance. So there is a correction over here. So it can be cross resistance or it can be multiple resistance. In this example, what I told you, it is multiple resistance. Say, take the case of 2-pyrethroid. You have used deltamethrin continuously. There will be high level of esterase enzyme. Then you have used cypermethrin. Again, there is an ester bond. It will break ester bond. So there will be cross resistance. So this detoxification can lead to development of resistance. Now let's see one by one. First, we will see esterases. That's the one which we are discussing. It's a ubiquitous enzyme. What's the name by ubiquitous? What's the dictionary meaning of ubiquitous?
Present everywhere.
Yes, present
Everywhere.
everywhere. It is present in all organism, and it is involved in the degradation and metabolism of chemicals. So it can be phytochemicals, it can be pesticide, any we call it as xenobiotics, any other foreign compound. Xenobiotic, I use, sometimes I may use a term called a xenobiotic. Xenobiotic is nothing but a foreign chemical. Okay. And this esterase, as I told you, it has the ability to hydrolyze ester bond, amide bond, as well as thioester bond. So, esterases not only catalyzes the metabolism of compounds containing ester bond, it also catalyzes the detoxification of compounds containing amide or thioester bonds. So, this enzyme can detoxify pyrethroids, OP compounds, as well as carbamates because these three groups are having ester bonds. And most of the resistant insects have higher levels of esterase enzyme. They overproduce the ester. So what we normally measure is that experiment was also conducted during the analysis thesis work. But what we did was we first did the classical bioassay. We found out the whether the insect has evolved resistance or not. Then we once we detected resistance, we went for this biochemical mechanisms to identify which enzyme is contributing to resistance, and we found out that esterase level of esterase enzymes is more in the case of resistant insect compared to susceptible one. We measure esterase level in resistant strain as well as susceptible strain. We'll compare. The resistant strain will have more level of esterase enzyme. That's how it develops resistance. And how this level of esterase increases. What is the mechanism and what is the mechanism of detoxification? One mechanism of increase in detoxification level is gene amplification and upregulation. Gene amplification, as we have seen in the earlier picture, there we have seen instead of the susceptible individual was having only a copy of the gene, but the resistant individuals were having 4 copies of the gene. I will give an example of again an mosquito. You can see it over here. In Culex, you can see in the resistant individual were having 250 times the number of copy of the gene. That means if the susceptible Culex mosquito was having a single copy of the gene coding for esterase enzyme, the resistant individual were having 250 copies of the gene, same gene coding for esterase enzyme. And this increase in the copy of the gene number led to 500-fold increase in the level of enzyme. And with this 500-fold increase in the enzyme level, the insect developed 800-fold resistance to OP. Can you now imagine how resistance occurs? It just multiplies, it makes more and more copies of the gene. Esterase gene is always present in insect, no doubt. What it will do? It has to produce more quantity of esterase to detoxify this pesticide. So what it will do? It will make more copies of the gene which will code for this enzyme and see the result. And another mechanism what the insect will do is here the sequestration. That means the enzyme will sequester the pesticide. What is the literal meaning of sequestration?
Assimilation.
Yes.
Arranging.
No. Sequestration is nothing but binding. You can consider sequestration. If once sequestered means it means it is bound to the literal meaning is binding. So that means enzyme will remain tightly bound to that insecticide. So what will happen? The insecticide actually, if the insecticide has to act, it has to bind to acetylcholinesterase enzyme. But instead, it will bind to another esterase molecule other than acetylcholinesterase. And the insecticide won't be available for interacting with acetylcholinesterase. So the normal activity of insect can continue. So it binds to the insecticide and the insecticide won't be available for reacting with acetylcholinesterase. And the second one, that's what I told you, mutations. If insect has to develop more copies, it has to mutate, then only it will develop more number of copies. So that mutation can be by gene amplification. And another thing which I didn't touch up is upregulation. Instead of increasing the number of copies, have you heard studied about upregulation, downregulation in the biotechnology course, molecular biology course, UG? Yes or no?
No, sir.
No. Upregulation, have you heard about promotion of gene expression promoters and all?
Yes, sir.
Yes, yes.
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 So, yes. So in upregulation, what happen is there will be a set of sequence of genes which regulate the expression of the gene. So there is a gene coding for production of esterase enzyme. So the before the in the chromosome, if you see that gene, particularly that gene which is coding for esterase enzyme, before that enzyme there will be a set of nucleotides, a set of enzyme which will control the expression of this enzyme, the gene coding for estrogen. So what it will do is this set of enzymes will make this gene coding for estrase enzyme to work harder or overexpress. That means that gene coding for estrase enzyme, there will be the gene which is regulating the expression of the gene coding for estrase enzyme. It will tell the gene estrase gene to work harder, express more. That means work hard means more production of estrase enzyme. So without increasing the copy of the gene, what it will do is it will increase the production of the gene. It will that upregulation of genes, estrase gene, will lead to more and more production of esterase enzyme. So even without increasing the copy of the gene also, the gene will express more and will lead to the overproduction of enzyme. Understood? This to what is gene amplification and upregulation.
Yes, sir.
Yes. And
The
if you see,
promoter.
ah,
Promoters
Without from
with it controlling
promoters, the function of promoters is to whether to determine whether the gene is expressed or not, isn't it?
yes,
Only
sir.
if a promoter gene is present, the gene will be expressed, isn't it?
Yes,
Yes
sir.
or no? Yes,
Yes, sir.
that's the role of a promoter. Say, consider promoters can be considered as one upregulation of gene. So, if the promoter asks the estrase gene to work more, then estrase gene automatically works more and more. Instead of working say 3 hours in a day, it will work for 6 hours. I'm just joking. It may work for 6 hours, 12 hours, 24 hours a day. So if it works for 24 hours a day, more production of enzyme estrase. Okay.
Okay, sir.
So what and how this upregulation and all those things happen? In the normal scenario, the promoter will give a command in a routine manner. In a normal scenario, it doesn't command the estrase gene to work more. But here, there can be some mutations which will lead to the changed upregulation of gene. Take the case of one typical example that is again seen in mosquito. I forgot to give the insect. Here, have you ever heard about this kind of expression? GLY 137 ASP or TRP 251 LEU.
Corona.
Yes. Uh, what Corona what they do with this kind of expressions? This is concerned with mutation. Actually, what would happen is that it 137th position. Earlier, it was GLY is the abbreviation of the amino acid glycine. There is a universally accepted abbreviation for each amino acid. At 137th amino acid, earlier it was glycine, and in the mutation, because of the mutation at 137th position, now it's not glycine, now it's asparagine. ASP is asparagine. Aspartic acid is different. So in at 137th position, in the normal individual, it will be glycine, but in the mutated individual, it will be the amino acid will be asparagine. And because of this mutation, the gene estrase gene will be overexpressed. And in some other insects, at 250 was position, in susceptible individuals it was tryptophan, but in resistant individual it got mutated to leucine. So this mutation can regulate the expression of it can lead to the changed expression of the gene, the estrase gene. Clear?
Yes, sir.
One thing, if you you go back to a biochemistry and molecular biology part, or just Google it. If you see the nucleotide sequence for glycine and asparagine, it will be only. I will just see. I will just check it. Glycine. I mean, mm, in the next class, I will show you how it is. You might have studied in different ATGC, which are sequences which contribute to glycine, the trinucleotide sequences. Just a minute. I will tell you later. Okay. Now it takes some time. I have to find it out. Okay, because if you see what I have to wanted to tell you is that if you see glycine and asparagine, it will differ only in one nucleotide. So a single change in nucleotide will convert glycine to asparagine. So that's why we call it as point mutation. A single nucleotide will get mutated, it will converted to another nucleotide. So the entire amino acid will get changed. Instead of lysine, it will be asparagine. That's how that mutation occurs. Coming to the next one, that is glutathione transferase. Actually, this glutathione transferase, one thing we have to remember is I will take after resistance, I will either I will take metabolism of insecticide. In that metabolism of insecticide, again, I will discuss about glutathione transferase. If the in the metabolism of insecticide, if the insecticide has to be metabolized quickly, it has to be made more hydrophilic so that it can be excreted through water. So this is one more enzyme. Glutathione transferase is one enzyme which catalyze the reactions which will make the enzyme, which make the insecticide more hydrophilic, so that it can be easily excreted. So this glutathione transferase is a large group of enzymes, and it is detoxifying large group of toxicants. So their main function, every organism has a molecule called as reduced glutathione. You can see it over here, GSH. GSH is nothing but reduced glutathione. As you can see, there is a thiol groups, there is a sulfur group. So this glutathione transferase will catalyze the reaction of the pesticide molecule or any xenobiotic with reduced glutathione. So this reaction, this is a conjugation reaction, that means binding reaction. So reduced glutathione will conjugate with pesticide. Once this reduced glutathione is conjugated with the pesticide molecule will become more water soluble or hydrophilic so that it can be easily excreted. So the glutathione transferase is an enzyme which catalyzes conjugation reaction. Actually, this conjugation is between reduced glutathione and xenobiotic. We take the here we can take the case of pesticide. So once the pesticide reacts with or conjugates with reduced glutathione, becomes more water soluble and it can be easily excreted. This is mainly involved in the resistance of organochlorines, and because it leads to dehydrochlorination reactions. It catalyzes dehydrochlorination reactions. So it is involved in the resistance to organochlorines, organophosphates, as well as pyrethroids. Other groups also, it is literature says it is also involved, but mainly with respect to organophosphates, organochlorines and pyrethroids. Again, similar to esterases, again this resistance is mainly by gene amplification or gene duplication or upregulation of gene. Upregulation of gene or gene duplication or gene amplification are the mechanism here also, just like esterase. Now let's move on to the last group, that is mixed function oxidases or cytochrome P450 monooxygenases.
Sir,
Yes.
sir, I'm having classes. So,
Okay, you can. I will stop. You can leave.
okay, sir. Thank you, sir.
I'll take tomorrow. Okay, then